Okay. Good morning, everyone. I want to welcome you all here today in this kind of wintry weather to our December MP seminar. I have a few announcements before we get started. One that's of particular importance to the crowd. The January seminar has been canceled. One scheduled for January 21st, but the February one is still on tap. Located here, PRV 105, start at 8:30 a.m. And the speaker is Ranu Jung, PhD, Associate Professor, School of Biological and Health Systems Engineering, the Ira A. Fulton Schools of Engineering, Arizona State University. The title is still to be determined, but that information will be sent out via email as soon as we get it. I have another announcement to make. It gives me great pleasure to let all you people know that we have an award winner amongst our midst, Miss Allison Hess here, PhD candidate in electrical engineering and computer science, and an advisee of myself, won Best Poster Award at the MRS Fall 2010 meeting for the for her paper titled Fabrication and Characterization of MEMS-Based Structures from a Bio-Inspired Chemo-Responsive Polymer Nanocomposite. Yes, that nanocomposite that we've been working on with Dustin Tyler's group and Stuart Rowan's group and Christoph Bader's group for some time. I should say that there were 4 awardees out of about 500 posters, so that's a real accomplishment. Are there any other announcements that anyone would like to make before we go ahead and get started? Put my glasses on to see. Seeing none, let me move on to introduce our speaker. It gives me even greater pleasure to introduce today's seminar speaker, Heidi Martin. She's really one of us. She received her BS degree in chemistry and her BS degree in chemical engineering from Case Western Reserve University in 1993, followed by an MS in chemical engineering in 1996 and a PhD in chemical engineering in the year 2000, where she studied under John Angus, developing diamond, CVD diamond techniques, and all the great things that Case Western Reserve is noted for in the diamond community. After she received her PhD, she did a 2-year stint at the University of North Carolina, Chapel Hill as a postdoctoral fellow in chemistry and neurosciences, studying under Mark Whiteman. We were fortunate to get her back here after that postdoc stint, where she joined the Faculty of Chemical Engineering in 2002 as an assistant professor. Recently promoted to associate professor with tenure in 2010. Among her many accomplishments include the Case Alumni Association Meritorious Service Award. She was a Glennon Fellow in 2006 here at Case Western Reserve University. She held the Eric T. Nord Assistant Professor of Electrical Engineering Endowed Chair from 2002 to 2005. And she received the NIH National Research Service Award from 2000 to 2002. She's well known in the diamond community for her work in diamond as an electrochemical sensing material, and I'm sure that at least a good portion of her talk is going to be along those lines. She has moved that focus area towards neurointerfacing, and I really like that because she and I have had a very fruitful collaboration merging diamond technologies with polymer technologies to make microsystems that has the best of both worlds. We can talk about that another time. This time, Heidi Martin.
All right, good morning. We all set with the microphone? Okay, great. Thanks, Chris, for the great introduction, and to you and to the rest of the organizing committee from the FES Center and the APT Center for your kind invitation to speak to you today. As Chris mentioned, I've had a history of working with diamond, which is definitely a great conversation starter. When you tell people you grow diamond, you start getting people around you in droves. Our interest has been in the development of conductive diamond as an electrode material. And for this talk today, I'll be discussing that application in the area of neurological devices. So when I get started, first let's talk about diamond. Another well-known blonde once said that diamonds are a girl's best friend. And indeed, this is usually our first impression, what we think about when we think about diamond, that nice, very clear gemstone that's used in lots of jewelry. And actually, you can have not just clear diamonds, but you can have colored diamonds, which are usually even more expensive. And the way you get color in your diamond is by incorporating impurities into the lattice. In this case, you put boron in the lattice to make the diamond blue, this being the most famous of blue diamonds, the Hope diamond from the Smithsonian. And not only does the boron change the color of the diamond, but it also changes the properties of the diamond and makes it electrically conductive, which is of great deal of interest to me. Now, these are, of course, natural diamonds that you would see mined from South African diamond mines, but there are actually even companies now that are growing gemstone single crystal diamonds and are competing with De Beers in the gemstone market. And so they can grow diamond actually relatively rapidly and grow it as a single crystal, so it is real diamond, it's not cubic zirconia, it's real diamond of even superior quality of what you can get out of a diamond mine, and at costs, of course, that are definitely marketable. I don't grow gemstone large diamonds, so I don't want you all coming to my lab looking for them. Instead, what I do is grow diamond as a film, different kind of film than Marilyn was involved with. But our method of growth is chemical vapor deposition, which is a vacuum method where you start out with a carbon-containing gas as what's going to be your source of diamond. The traditional route involves that carbon gas being diluted in a hydrogen atmosphere, and you activate the gas to break it down, and you're under the appropriate conditions to get diamond growth instead of other types of carbon like graphitic growth onto a substrate. This is, again, a thin film. You can grow a single crystal film like the larger single crystal diamonds, but you can usually only do that on diamond itself. What you tend to do is grow a polycrystalline film where you grow lots of little diamonds that coalesce to form a film, and there's not anything telling these little crystals to grow all in the same direction, so you end up with grain boundaries between them, as you see here. Our growth rates are much slower, so there's no way that I can grow a very large diamond. It would take me months to grow something large enough that you could hold in your hand, the equivalent of something that would go into a ring, and it may not look that good at that point. Here's an example of a polycrystalline film. This is microcrystalline because the grain size is on the order of microns. And this particular film is a boron-doped film, which on a microscope doesn't really look any different. If you were to grow it single crystal, it will look blue because of the boron in that lattice. So diamond, as an engineering material, does have several interesting applications. Of course, one thinks of diamond as being very hard, and so ultra-hard diamond coatings have been even commercially developed, coatings of tool bits and saw blades for many years. There's also, diamond has the largest thermal conductivity at room temperature of any material, so there's interest in it as a heat sink, not as a doped material, but as a pure insulating diamond material. And then the doped diamond as an electrode material. Of course, my interest here is in the area of neuroscience, and this rather simplistic picture I'm not going to explain to you because this is definitely the audience that knows more about this than I do. But what I want to point out is that we're interested in using diamond in several ways in neural interfacing. And when we think about neurotransmission, we have both electrical and chemical transmission of information, as well as the idea of stimulating the neural activity to begin with. And we believe diamond would be useful for both detection of electrical signals and chemical signals, as well as stimulation of the neurons. The process of neurotransmission is very rapid in terms of being on the order of milliseconds. And in terms of electrochemistry, which you are going to hear some about from me today, you can do electrochemistry on this time scale. which makes it very useful for maybe monitoring things on the time scale of the neuron firing. So using diamond for electrochemistry, it actually is an expanding area, because diamond for electrochemical applications only requires the use of a thin film. You don't need a large bulk diamond to use it. There are several advantageous properties which I will come back to over the course of the talk, those being the wide potential window, a low baseline current, and minimized oxygen reduction, which is definitely very important in tissue applications because of the prevalence of oxygen in the tissue. And then last but not least, at least for the area of course, chronic implants, is the idea that diamond is chemically robust. Another reason that the use of diamond for electrochemistry is expanding is there are several different types of diamond materials now available. We originally started out making microcrystalline. There's also a couple variations on nanocrystalline diamond. This last one, ultra nanocrystalline diamond, involves a different type of gas environment. Instead of hydrogen, they use an argon-based plasma system to make the nanocrystalline films. In terms of applications, there are several length scales you can cover with diamond electrodes. There's companies in Europe that are making macro large electrodes for water treatment applications. There's interest in robust diamond electrodes for electroanalysis. And then, of course, if we go to the opposite extreme of the smallest electrodes, micro and maybe nano electrodes at some point for biomedical applications. So in terms of the advantages of diamond as a bioelectrochemical material, for diamond as a neurosensor, and again we're talking about in vivo detection for both chemical recording and electrical recording, the number one advantage is this idea of long-term stability. There's also, as I'll show you, the potential of improved sensitivity for chemical recording or electrical recording because of what we call the low baseline current of the electrode. The wide potential window, as you'll see, will allow us to maybe look at chemicals that we couldn't detect before. And I already mentioned about the lack of oxygen reduction. The diamond surface is also more homogeneous than other forms of carbon and also less reactive than metal surfaces, and thus makes it, I'm not going to show you necessarily a lot of data on this, but there's definitely some evidence that we can get more predictable surface functionalization as we need to for improving selectivity in terms of chemical recording. For neural stimulation, the stability of the electrode, the lack of reactivity of the electrode in electrolytes in general may help us to avoid reactions that occur at other electrodes that may be implicated in tissue damage. And that is one of, I think, one of the possible big advantages of diamond for neural stimulation. Also, because of some of the lack of these reactions that you don't see on diamond, we may be able to use diamond as a tool to help us understand neural stimulation from an electrochemical standpoint, which I think is very important. In terms of the electrochemical standpoint, both neural sensing and neural stimulation, I say neural sensing in terms of neurochemical sensing here, they do have an electrochemical basis. For neural sensing on the left here, you are applying either a voltage or a current to an electrode. It's a microelectrode on the order of 10 microns or so. We do, of course, like the size because of course, the spatial resolution you can get, but the currents are small, which should then reduce the idea of tissue damage from high amounts of current. And the size of the electrode also changes the mass transfer to the electrode, which allows you to use it in a faster time resolution, so you can get down to that millisecond time resolution to monitor things in real time as the neuron is activated. For chemical recording specifically, carbon fiber electrodes and metal electrodes are being used primarily for acute electrode studies. There are no chronic implants that are available, but there's a lot of work been done on chemical recording. My postdoc mentor, Mark Whiteman, was he is the, I guess, pretty much the inventor of using carbon fiber microelectrodes for neurochemical detection. For neural stimulation, which you guys, this group of course knows a lot about, is with biphasic high-frequency electric pulses. From the electrode's perspective, again, this is an electrochemical process. Ideally, we don't want any species, net species, added to our electrolyte, to the tissue. And thus, that charge injection must be a capacitive mechanism that is completely reversible. But there are possible electrochemical reactions called Faradaic charge transfer reactions where species can get introduced or removed from the electrolyte. And these are some examples of those that can happen on metal electrodes. You can break down the water at sufficiently high potentials. Oxygen reduction can produce reactive species, for instance, superoxide. And also the electrode itself can oxidize to form an oxide layer that could weaken the structure. Or in the case of platinum, for instance, at sufficiently positive potentials, you can form chloride salts. So again, electrochemistry is important, and we hope Diamond can maybe improve on some aspects as well as help us understand these. So what I'm going to talk about is I've split this into two parts. The first half of this talk will talk about the potential of the properties of Diamond for its performance and stability for neurosensing and neural stimulation. I'll talk about the idea of chemical sensing, the sensitivity and stability, as well as what we have so far in understanding how it could perform as a stimulator. And tied in with the stimulation is trying to help us understand the factors that we need to look at for actually evaluating stimulating electrodes. And then the second half of the talk will focus on looking at our newest electrodes, which are actually have mechanical integrity, but they are flexible, which is definitely important for getting them to remain in one piece in tissue. So our initial electrodes were based off of trying to mimic the carbon fiber microelectrodes used in neurochemical sensing. These are capillary-based electrodes that are, you pull the capillary to a fine tip. In this case, they would start out with a fiber inside the capillary, and they'd pull it and form a disc electrode. We started out by tapering, just to show you these types of electrodes, tapering a wire and coating it with diamond, but then you have to insulate it afterward, which is not necessarily a very easy thing to do reproducibly. But if we mimic that carbon fiber microelectrode, we can put a tungsten wire inside the capillary and pull it and create a little disc electrode. And then we put it into our CVD diamond reactor, and we can grow diamonds selectively at the tip. This is a 30 micron or 30 to 35 micron diameter disc electrode. Again, it's polycrystalline, as I mentioned earlier, and you coat it on various materials. These, I want to point out, again, are inflexible microneedle electrodes, so I'm not necessarily aiming to make them something that would be flexible to go in tissue yet, although we were hoping they would still work in tissue. So with these electrodes, what I'm going to focus on first is the electrochemical sensing, the neurochemical sensing. And in order to do that, just because of the diverse audience, I thought I would at least tell you a little bit about the method that we use. So in electrochemistry, what you're doing is applying, you have to apply an electrode potential to cause chemical reactions to occur. It's the equivalent of giving it the energy of the reaction. You have to cross a certain threshold of energy for a reaction to occur, and that's the potential. One of the methods that's widely used is what we call linear sweep voltammetry, where you control the potential and measure the current. And linear sweep voltammetry, this is what it would look like over time, sweeping the potential between two limits over and over again, and you monitor the current, and this current is associated with the various capacitive and Faradaic reactions occurring at the electrode. This, in this case, is the breakdown of water at the limit of your potential window. And this current is proportional to the concentration of species that have undergone charge transfer. if we don't usually plot the voltammetry in this manner, we tend to plot the voltage versus the current by, I guess, folding the current profile in half, so that now you have this voltammogram on the right where you have current on the Y scale and voltage on the X scale. And we have the limits of the window being where the water, in this case, this was a, I think, sulfuric acid solution. This is a diamond electrode where you get water breaking down. So I'm going to show you several of these. And so anything that occurs within this potential range, you'll be able to see it on your electrode. The voltammogram in that way acts like a fingerprint for what's going on electrochemically in your system. So again, here's that voltammogram for diamond. And what I wanted to point out is it does have a very large potential window. Platinum's window is only about 1.5 volts for comparison, and I'll show you that here in a few minutes for purposes of the neural stimulation part. The low baseline current here, I'm not showing you for a comparison, but its baseline is probably about 10 times lower than other forms of other metals and carbon-based electrodes, which should increase sensitivity. And this was a scan, 10 hours of cycling. It didn't really change anything on the electrode. So that was a general idea of voltammetry. For neurochemical sensing, you have to speed up the scan rate to be on the order of hundreds of volts a second. This particular piece of data was, I think, taken at 300 volts a second. And that means that that entire scan is only going to take you about 10 milliseconds. And so thus we can actually do voltammetry during a single neuronal, I guess, event. And what we see here, this is actually for detection of dopamine. Here's one of those carbon fiber microelectrodes I mentioned. There's the carbon disc that's at the end of the capillary. And here's the linear sweep. We've crossed the appropriate voltage to get dopamine, in this case, to oxidize. And this is a reversible process. So you see I have background current is the black scan. I add dopamine to my system. I get a little blip here in the appropriate potential region, which is associated with dopamine being oxidized and the reverse reaction, the quinone being reduced back to dopamine. And we could subtract out the background, see the current, and it is proportional to the amount of dopamine that's present. So with those diamond microdisc electrodes I showed you, here's an example of our sensitivity for dopamine oxidation. This was 1 nanomolar dopamine, which of course people know is physiologically relevant. This is what we call a false color plot, where we've run that linear sweep voltammogram many, many times over the course of this 15 seconds, and each vertical line in my plot is one of those scans. And so what we can do is then look at the voltammetry over time, and whenever we see a change in the color, that means the current at that particular spot has changed. So if we were to take a slice at any given potential over time, you can see the current for dopamine go up and come back down. In terms of what that current corresponds to, you have to look at the voltammogram directly and identify the particular peaks which have been independently identified as dopamine oxidizing and dopamine being reduced. This particular voltammogram, a couple I have, are switched in terms of the potential. I have the positive potential on this side in this particular scan. They often are, or most of the time, depicted in the opposite direction. In terms of the stability of this response, for a larger concentration of dopamine, here's 10 micromolar dopamine. The purple scan is the oxidation peak, injected about 50 times. My student went home, got some sleep for some reason, came back, and he didn't have to polish the electrode, rinse the electrode thoroughly, do any kind of cleaning. He put it back in the electrolyte, and he got the dopamine signal right back, which this is, of course, just over a couple of days. There's been people that have reported stabilities, weeks and months of various chemical detection. So this is the idea of diamond being chemically stable. Now, that 1 nanomolar signal that I showed you before, though, unfortunately, we haven't been able to keep that stable for some reason, and of course that's very frustrating for me to understand why that hasn't happened. But the good news is I think we're back at that level with some of our newer electrodes, so all is not lost there. But anyway, to point out about the stability of at least the larger concentrations, it's interesting that both the oxidation peak and the reduction peak are stable, which is unusual. Some electrodes don't have the good stability of the reduction peak. That reduction peak becomes an advantage when you look at chemical selectivity. This is two examples of dopamine. I'm sorry I didn't write out the words here. Dopamine and ascorbic acid, which these are in appropriate ratios of what you'd see possibly in tissue. The problem here is that ascorbic acid also oxidizes at the same potential as dopamine, and that's right here happening. And so if you're trying to detect dopamine in the presence of ascorbic acid, you're never going to be able to tell what the concentration is. But dopamine has a reduction peak, again, well defined on diamond. Ascorbic acid doesn't have one, so you can see in a mixture of ascorbic acid what the concentration of dopamine is by using the reduction peak. Also, serotonin and dopamine oxidize the same potential because they're very chemically similar molecules, and consequently, again, you couldn't see dopamine with serotonin present, but the reduction peaks are located at different potentials. I don't show the plot, but I do have data showing similar to ascorbic acid, where you can do a calibration of dopamine in the presence of serotonin. So that's one way we can get selectivity. You can also do surface functionalization, which is, there's several different ways to do that. So even though diamond is chemically robust, you can get surface modification. It's usually a free radical mechanism. This is one example of that, using a diazonium compound, which you generate a free radical by eliminating this nitrogen on the end of the phenyl group, and you can attach various substituted phenyl groups to your surface. This particular molecule would be more electronegative on your surface, which would repel ascorbic acid. And so before, we had a problem where dopamine and ascorbic acid were oxidizing in the same place. But once you add the sulfophenyl to the diamond surface, the blue scan here is the ascorbic acid signal on the diamond, and we've pretty much wiped it out. Versus the dopamine signal, actually the blue scan was after the surface modification here as well. We could see the dopamine just as well. So we have the option to either look at the reduction peaks and use that as a way for selectivity. We can also explore chemical functionalization. We have used these microdisc electrodes in tissue. This is an in vitro study I've been doing with Chris Wilson from the pediatrics department here at Case and Rainbow's Babies and Children's Hospital. What we were doing is looking at the concentration of the neuromodulator adenosine in the pre-Betzinger complex, which is in the brainstem. They believe that this is the site of the neural circuit controlling respiratory rhythm, and that adenosine plays a neuromodulatory role in that circuit. So what we did here was we put a diamond electrode into a brain slice of a rat pup, and this particular experiment was to show we could see adenosine. This is important because adenosine oxidizes at a high positive potential, higher than is possible to see on other electrodes. So here for the first time we could directly measure adenosine in tissue. And again, here's one of those false color plots where we've injected this molecule called CGS there was a number after it I left off. So I apologize for not being complete there. But when we inject this, it's an agonist. It should enhance the release of adenosine in the tissue. And we do see a color change in the appropriate spot. You just have to believe me that that's where adenosine oxidizes. And we see the concentration, this is background subtracted, we see the concentration go up. We did several experiments. Here's actually the Voltammogram showing the oxidation peak for adenosine. And we did several different types of experiments to try to verify we were seeing adenosine in the tissue, bath administration versus local administration of the agonist. We also put a blocker in and saw the concentration go down. So we were relatively confident that we were seeing adenosine. Dopamine oxidizes at a lower potential, so it's not going to interfere with adenosine measurement.
Before you leave that, could you back up there?
Yeah.
For I don't understand that scale on the upper left panel, minus 1 volt.
Oh, this?
Yeah, what's...
Each one of these vertical lines is a complete voltammetry sweep. So I've started at -1 and gone up to 1.5 and back down to -1. So I've completely done the full scan in both directions. So it's not just in one direction. So if I were to go back to the idea of the linear sweep, I've actually done the whole cycle. And I'm just showing each one of those vertical lines is one of these cycles at taking 10 milliseconds at a time. Okay? So it's a compact way of showing a lot of voltammetry data. Does that answer your question?
I guess I don't understand. It's probably okay. I just don't understand why -1 is at the top and -1 is at the bottom.
Oh, well, I mean, imagine that the potential's going up and coming back down. So, I mean, I could have shown half of this, but it's to try to show the whole sweep. I guess we'll have to talk about it more later. I know where your office is. So, actually, you know where my, and you,
learner I am, so.
You know where my office is too, for that matter. So, okay, so anyway, we believe that we were able to see adenosine, and what we're trying to do is actually, we're hoping to continue this and start looking at more relevant questions in terms of the role of adenosine. But what I wanted to point out is that because dopamine does not oxidize at the same potential, we can actually see them both at the same time on the same diamond electrode. And this was some data from a slice, and this is all background subtracted. So it's subtracted from the data taken before you applied the agonist. And consequently, the current went up for oxidation of adenosine, and that's shown down here. Sorry, do that one first. So it went up before, like I showed you in the last slide. The dopamine concentration, there's a lower potential for oxidation. It went down. That's why it's shown as a negative peak, even though it's an oxidation, because it's relative to the background level, and that's shown here. So it's interesting that we can see these both simultaneously. Okay, so hopefully I've given you a little indication about using diamond for neurochemical sensing and what neurochemical sensing in general is about. Let me turn to talking about stimulation. And of course, for a neural stimulating electrode, besides it being, of course, biocompatible, we need it to be corrosion resistant and have the sufficient amount of charge, a current for charge injection, and of course not have that tissue damage, which from an electrochemical standpoint means lack of irreversible charge transfer. And of course, platinum is most commonly used. There's possible issues with it corroding or producing some other species. We believe diamond may be able to help in that regard because of its stability. Here's just another example of one of those polycrystalline films on a wire, which we're using for these studies. And with that, along with what I'm showing you in terms of comparing these two materials a little bit, is we're also trying to, with Tom Mortimer, who was one that just asked the question, we want to be able to try to understand the electrochemistry of the stimulating electrodes so we can then hopefully understand how damage occurs to the electrode or to the tissue. So in terms of doing this, we had to develop a language, or I did at least, because I'm not a neuroscientist, of how electrodes operate for stimulation and what we actually want them to do. So we need a stable electrode that reversibly injects a large amount of charge. And one of the methods that's been reported in the literature for preliminary evaluation of stimulating electrodes is an electrochemical method called slow scan voltammetry, which you've already seen in the context of neurochemical sensing. But in the case of neural stimulation, evaluating these electrodes, what they do is to integrate the charge under the Voltammogram, since charge is the integration of the current over time, and they define something called the charge storage capacity. And if the amount of charge in the cathodic direction is the same as the charge in the anodic direction, then the reversibility is high, which means nothing new is introduced into the electrolyte and thus into the tissue. There are several factors to consider because it's an electrochemical process. Dissolved oxygen is there, so I'm going to show you a little bit of data related to that. And of course, that could be important related to oxygen radicals. There is significant, of course, ionic and organic content in the tissue, which may result in fouling, may affect the actual performance of the electrode. And then just in general, because they're using this slow electrochemical technique for evaluating electrodes preliminarily, what happens when we, compare that to the actual more relevant time scale, that fast pulsing situation. Okay. So looking at the first of those in terms of oxygen reduction. So here's a comparison of the voltammograms of diamond and platinum in phosphate buffered saline. This shows the activity, and I hadn't shown you this yet. Here's the voltammogram for platinum, and here was that voltammogram for diamond, which looked actually relatively unexciting because it's just a line. But for platinum, there's a lot going on here because you get adsorption and desorption of hydrogen. You get the oxide of the metal forming and getting reduced. This is in a nitrogen environment, this dark scan. If you add oxygen to the tissue, you get significant oxygen reduction. And actually, platinum is a very common metal for, as a catalytic electrode for oxygen reduction in, say, fuel cell applications. So it definitely is very good at reducing oxygen. Diamond, on the other hand, doesn't show all this behavior of hydrogen adsorption. There's no oxide formed. There is, because there's a polycrystalline material here, there's a little bit of grain boundary activity, and there is some minor amount of oxygen reduction, but not nearly as significant as on platinum. So what that means, if I calculate the charge storage capacity, which might predict how much charge that it's going to inject and also how reversibly the charge will be injected, this is platinum. And what you have here is, here's the cathodic charge, so going in this direction, going negative. All the current that's below 0 would be the cathodic charge. If we look at the positive charge, is all the anodic charge going in the positive potential region, current that's above 0. And those are pretty well equal in a nitrogen-saturated electrolyte, PBS. Of course, that's not the way tissue is. Tissue has some oxygen present. This is the extreme case of saturating it with oxygen, which of course is much more oxygen that's in tissue. But in that case, you see the significant cathodic reaction of oxygen reduction here. The anodic situation doesn't really change much, so the net amount of current is very negative. So the total amount of charge transfer here is quite irreversible. In diamond, it doesn't seem to matter if the oxygen is there. The currents are actually in the same order of magnitude as the platinum. The net amount is low, so it's very high reversibility. And again, the same is for oxygen. Now let's turn to organic content. We all know that there's proteins in the tissue, right? Lots of stuff in there that can gunk up electrodes. And how does that affect charge transfer? And again, we like to think of that picture from the last slide, this beautiful voltammogram with lots of clearly defined activity, which will contribute to charge injection for stimulation. But when you go to an electrolyte that has a significant amount of proteins, we've done a few different electrolytes. One of those is actual hemolymph from the Aplysia californica from Hillel Chiel's lab here in biology. This is the extracellular fluid and also the blood of the animal, so a lot of proteins present. If we compare that to phosphate buffered saline that just has ionic content, the voltammogram is a lot different for platinum. We lose a lot of that activity for hydrogen adsorption. The amount of current overall is down. And so that will affect how charge transfer and this charge injection would occur. On diamond, it really didn't change. Again, there's not really, I'd have to show them on the same page, but this is the same as we saw before. We did expand this a little bit to show a little bit of activity from some kind of organic content, but again, not affected as much like platinum. Okay, so that is voltammetry of diamond and platinum related to neural stimulation. But of course, we're interested in getting to conditions where we are simulating neural stimulation, the high frequency pulsing. And so with that, in collaboration with Tom Mortimer, we've started using a custom instrument that would hopefully try to mimic a biphasic balanced charge stimulation pulse. And this is called a current pulse capacitor discharge instrument. And we're using a custom instrument because a lot of, or every commercial instrument that we've looked at so far has had a significant amount of leakage current. And if you're wanting to understand things from an electrochemical standpoint, we can't have this extra leakage current that is interfering with our interpretation of our data. So what we have here is basically we charge up a capacitor and then we close a switch to discharge the capacitor. And that's shown here. And the amount of charge that gets discharged should be, of course, equivalent to what you put into it. So it does imitate the idea of a balanced charge pulse. The potential, the other thing about this instrument that is good is we can monitor the potential of the electrode. And thus we can see within that potential window, that fingerprint of electrochemical activity, where our electrode could be sitting and thus what could be chemically happening at our electrode. In this case, it shows an example where the potential went more positive than it originally started at after the pulse. And that's an indication that the electrochemistry was not completely reversible. If it was reversible, it would have come right back to the same spot. So again, that's what I was trying to say. The potential increase indicates irreversible transfer. And this just shows the idea. I didn't really explain this. This is the instrument here. This on the right is a model of what an electrochemical cell looks like with your, in this case, platinum or diamond electrode. It would be, you have to have a counter electrode, and there's a reference electrode so you can monitor the potential. And then there's various capacitances and resistances in your circuit corresponding to the actual physical phenomenon. Okay, so here's some results from that. And again, this is now a 100 microsecond pulse. This is, of course, much more relevant for stimulation purposes. We've put the Voltammogram, taken the slow scan Voltammogram, which is taken on the order of seconds, for a comparison to show you where we are in that potential window. This is that idea of, this is actually a narrow window where, again, you don't see as much of that hydrogen adsorption and desorption that you'd see on a typical platinum electrode in PBS. And when we introduce this pulse, we do see the potential go higher, not by too much. This is a nitrogen, I should say this, yeah, nitrogen purged electrolyte. It only goes up, on the order of 10 millivolts after a single pulse. So again, but that's nitrogen environment. You add oxygen to the system, the Voltammogram will look different. Again, we're sweeping it with a current pulse. The potential ends up in this region here, goes back. It goes beyond, again, the open circuit voltage where we started. The voltage increase is more with oxygen present. So adding oxygen definitely on this time scale, just like what we saw with the Voltammogram with the charge storage capacity measurement, the oxygen decreased reversibility. With diamond, I won't show you both nitrogen and oxygen. I will just show you the oxygen case because it didn't really change much. Very little irreversibility in terms of the voltage being above the open circuit potential where we started. So we do believe that diamond could be possibly more reversible. At least this is what this is preliminary showing than platinum. What we need to work on, which I don't have time to show you, I don't really have too much data to show, but I don't have much time either. We do need to enhance the charge injection density at diamond, which we're trying to work on. One of the last thing to point out before I turn to my electrode, some other electrodes working on, is just comparing that time scale. Again, they're using voltammetry for preliminary evaluation, and that's on the order of seconds that you take the measurement. When you do the pulsing, you're doing something on the order of microseconds. And if you just compare the charge that gets transferred, this large amount of charge here is from the Voltammogram. And if you do the equivalent change in potential by applying a pulse, the amount of charge is much less. So by changing the time scale, which this is obviously the more relevant time scale, the amount of charge transfer you're seeing does seem to go down by quite a bit. And so maybe using voltammetry is not necessarily the best indicator of how much charge you're going to see at your electrode. Okay, so hopefully I still have you following me here. Got heavy into some electrochemistry. We've talked about properties of electrodes, and with the remaining time, I want to talk about some of the new ones we're making that are more flexible. One of the reasons we haven't gotten farther with the tissue measurements is because of the limitation in using those microdisc electrodes, because that they break. And this is not unusual. Here's my electrodes I was showing you, and we did do some in vitro measurements. This is most of the literature out there on making diamond microelectrodes, and most of them have made them in a similar way of making needle electrodes, and very few have been able to be used in tissue. This particular example here from Anaga's group, Suzuki et al., they did in vivo measurements because they took their electrode and broke off the brittle diamond end from the rest of the lead and then glued on a flexible metal lead to it. It's the only reason they could get it into the animal. So there's definitely a problem with the strategies being used for making these microelectrodes. And of course, that limitation is how we integrate diamond with other materials. And I should point out, of course, these are single electrodes that I was talking about here. There's also work on arrays, including my work with Chris Zorman here at Case. So one of the issues is that in order to make diamond more flexible, to be more compatible with tissue, we need to be able to integrate it with low temperature materials. And that's obviously more straightforward with metals because you can deposit them at room temperature. But diamond is deposited, those substrates, I hadn't mentioned at the beginning, that substrate is going to be about 800 degrees centigrade. So you're not going to be able to grow diamond directly on a polymer or a low temperature glass of any form. Instead, you have to think of other ways to get at this problem. There has been a little bit of work there's always been work at trying to lower the growth temperature of diamond, partially to reduce the cost of growing it, but there has not been really too much successful work on getting diamond onto a polymer directly. There has been recent data showing this nanocrystalline structure transferred over to various materials for heat sink applications, but those were not electrodes. Of course, we want that because of the biocompatibility and the flexibility. There has been one report of an all-diamond probe that looks like a shank electrode that is a combination of conductive and non-conductive diamond. And that is rather exciting, although it may be difficult to change, I guess, the overall dimensions of the electrode to accommodate what you need it for. So what we've done instead is to think about ways to get them flexible by either changing what we grow it on so that it stays ductile instead of being brittle, or this method of putting diamond on polymer. How much time do I have? Am I good? Okay. Okay. So the first method of putting diamond on a high temperature material that remains ductile, this goes back to the idea that those tungsten micro wires that we were growing on, those discs, in order to get good adhesion to that, there's a carbide layer that forms, and that carbide layer becomes very brittle. And for large electrodes, you can pretty well snap your electrode just by dropping it by 2 feet from a desk. If you drop it properly, you will break the electrode. And it's not because the diamond isn't hard, it's just because that substrate underneath has gotten to be so brittle. So we found a set of alloys that are alloyed with rhenium, which doesn't form much of a carbide. And we were able to grow diamond that would have the substrate remain more ductile. And the structure we are looking at is a hook electrode. This is for a project we're doing with Hillel Cheel in biology for wrapping electrodes around a nerve for electrical recording. And we preformed the hook because we're not trying to make the diamond film flexible. We're just trying to make the lead stay flexible so it doesn't break off. And so we're trying to replace the idea of this very cumbersome idea of taking a diamond electrode, breaking it off, putting a metal wire and gluing it together, globbing on some epoxy or something to make it work. Instead, we want the whole thing, including the lead, to be made out of one wire. And again, that application right now, although this is definitely what we're limited to, is for the aplysia. We're trying to wrap the electrode around a nerve. Here's buccal nerve 2, is associated with the feeding mechanism of the animal. And this was a first-generation electrode that we did that was not flexible yet, but we were able to implant the diamond electrode in a live aplysia and get neural recording while the animal was feeding. And this is just one example of a feeding pattern for actually a bite behavior. And that's about all I can tell you about bite behaviors. But in any case, we videotaped the animal. We're able to correlate the video with the electrical recording for several days, actually. I think the diamond electrode stayed in the aplysia for a couple weeks, and then the animal itself expired. So we're hoping to, again, move the ones that are more ductile, if we can get them into the live animal and do a similar thing. Those particular electrodes, we want to verify it still can oxidize and reduce dopamine. So we could think about this as a new substrate for making diamond for a neurochemical recording. We haven't insulated these yet, though, to use them. But what we wanted to show is, the whole point of this was to show that this new substrate is more flexible and thus will be more compatible with tissue. Here's our original substrate, tungsten. And the control here is just growing in hydrogen, where you're not going to get any diamond, and it should stay ductile, and it does, because the maximum of the test is 90 degrees that we bend it. And after 4 hours of diamond growth, that's this red bar here, tungsten breaks after 20 degrees, just snaps right apart. Tungsten, rhenium, molybdenum, rhenium. Tungsten, rhenium is better, but the best case is molybdenum, rhenium, where you have to sit there and bend it back and forth before you actually eventually get the substrate to break. So this is very exciting, because this could help not just for biological applications, but in general, this could be a new type of substrate for making diamond electrodes. And that goes back to the microstructure of why this works. The tungsten gets completely carburized. This is a brittle phase. Here shows a very ductile core of the molybdenum rhenium with a slight brittle phase on the outside. And again, this is an alloy, so the molybdenum may still form a carbide. Tungsten rhenium looks similar to this, but probably more carbide, because the tungsten is better at doing that. I'll skip this. This is the idea. We're also doing tension tests to do more mechanical testing. So the other strategy is, which is involved in putting diamond with a polymer. And again, I said we can't grow diamond directly on it. So what we've done is develop a transfer technique for doing this. And this, again, is with Chris Zorman and with award-winning Allison Hess and David Sabins on my side, who's a winner to me too, right? And so we have here some first prototypes. This is a diamond, 9 electrodes, or it's 9 pads as 1 electrode with a palladium contact. This one did not have, was not insulated, so we could not use it as an electrode. The palladium was still exposed, but it showed that we could transfer the diamond, and I'll show you what we actually do in a moment. This was a gold contact here on the right-hand side, where we actually insulated the gold. And we had to watch how this was sliced in order not to expose the gold for, again, for electrochemistry. The polymer we're using is polynorbornene. This actually fluoresces quite a bit. We wanted to analyze our diamond with Raman spectroscopy. We had to switch polymers in order to be able to see it in the Raman, and that is just an example. It doesn't seem to adhere as well in the Parylene. So what are the characteristics of this? One of the ideas here is, again, diamond is a brittle material, and if we use a large electrode, it may break. So we've got a design here that has a segment electrode approach, which has small pads that are combined to form possibly 1 larger electrode. The first generation we did was making individual electrodes at a time, where we were focusing more on the idea of transferring and not on all the microfab to incorporate the metal contacts, but we'll get to that. I better skip through this because I'm running out of time. But we have a selective diamond growth process developed by David Sabins, where we actually deposit the diamond with a metal mask present. We're depositing on silicon dioxide, and we remove the mask and then get selective diamond growth after that to form these pillars, which look something like this under confocal microscopy. So we're actually exposing 3 sides of the diamond that we're going to grab onto with our metal contacts and our polymer support to make have hopefully good adhesion to the diamond pads to form our new type of diamond on polymer electrode. And once we have the diamond pattern, then we build up the metal contacts here. That's shown as this darker yellow, and the lighter yellow is the polymer. This is done, of course, a series of microfabrication steps, which I can't go through now. The key here is that the polymer, polynorbornene, is resistant to hydrogen fluoride etch, which is what we're going to use to remove this purple layer, the silicon dioxide layer, to release the device and now get diamond onto the polymer. And the metal is, of course, hidden in here, so we have a true diamond electrode in a polymer. How does it act? Well, this is the first generation one of these. This was actually one on palladium that shows the dopamine oxidation. This is just 100 micromolar. This was a large electrode. It wasn't really small enough to do microelectrode work. The gold activity, if gold was, in this case, this is palladium, but I think this was a gold contact, not this palladium one shown here. But the gold signal, if it were exposed, you would see it, like a sore thumb, actually, in the Voltammogram. And this shows that we can bend this in the bottom of a cell. We've moved on, of course, to a much more microfabricated process where we're making 10 individually addressable electrodes. This is a segment electrode of 10 segments per electrode. And with that, we end up with these. We have 10 diamond pads here, each individually addressable with a nice bendable lead here, a chromium gold chromium trace. Mechanical properties of the polymer are not really impacted by having these very small diamond pads incorporated in them. This is just a picture of those showing the multiple layers. The diamond is optically transparent, and so is the polymer. So what you're seeing here is all the layers through at one time, even though the diamond is the only thing that's exposed on the electrode side, and nothing is exposed on the back except the polymer. That's kind of shown here. So here's the diamond side, and you can see here's the diamond popping up out of the polymer. And what's cool about this is where we flip the diamond over during this process, now the diamond surface is very smooth, reflecting that original surface where it grew on the silicon dioxide. And you can see that, you can sort of see where the electrode contact is underneath the polymer there. Okay. We had to work on the contacts. It's just, I like showing pictures to show the difference between what a good electrode and a bad electrode look like. The diamond, if it popped out, this is what you would see. It's a good thing to notice that the contact is ohmic. This is especially exciting because once we put the diamond onto the polymer, we can't do any high temperature processing to anneal our contacts to get ohmic contact like you typically do. Yeah. So in any case, we've got a working device. What we had to work out is the fact that the diamond surface we've exposed has some kind of insulating layer on it. This is shown by the fact that we don't really see any electrochemistry, we just see a capacitive response. This is from a cortical probe. And there's various parts of this device where you could have a problem. I already showed you we have ohmic contacts at the connection, so we don't think it's there. But what we wanted to look at is the surface. To make a long story short, we basically found that there's a silicon oxygen carbon layer on that interface, which is not entirely surprising based on the adhesion of diamond to silicon and how you have to form a carbide. That's reflected here by showing silicon and oxygen that we can etch through that there was a layer there. And that's shown there. We've actually etched that away with a fluorine plasma, gotten rid of the layer. There's a slight amount of contamination at the surface. The net result, and this is the final piece of data shown, is we now can use this for electrical sensing. This is a microelectrode, so we're able to do fast scan voltammetry. And this is not dopamine yet. This was a standard redox couple down to 100 picomolar using ferrocyanide oxidation in PBS. And so now we're back down to the levels we wanted to be at originally with our microdisc electrodes, and we're going to move to dopamine detection of neurochemical recording. So with that, I close. Thank you for your attention in terms of looking at diamond for neural stimulation, neural recording. I think it's unique for being able to be an integrated material for this application. But I think the key in order for us to move forward with implanting it is these new strategies of making diamond more flexible to be more compatible with tissue. With that, I acknowledge all the great people who've helped me, both all my students who've graduated in the past, Jeff and Song Tao, David Sabins, who just defended his PhD this semester, and Eric Hudak, who's soon to follow him, and Allison from Electrical Engineering and Computer Science, and all of my wonderful collaborators here at CASE and my funding sources. Thank you.
Thank you, Heidi. It was a delightful, wonderful talk. And as a token of our appreciation, the whole community here, we have these modest gifts which you may enjoy at a later date. And because right now what I want to do is move to questions. And so before we get started, we have a microphone that works, and we should utilize it if we at all possibly can. So if you have a question, either speak very loudly, because we think that the audio might not pick it up, or request a microphone. I think Dominique wants to open.
Right. So you talked a lot about the properties of these electrodes for stimulation, but you didn't say much about the recording. Do they have a lower impedance, for example, compared to metal, noise properties, and half-cell potentials? I'm interested in all the stability over time.
Oh, you mean for electrical recording?
Recording, yeah.
We haven't actually analyzed it too much for electrical recording yet. I mean, we've done some recording in the Aplysia, but we haven't done all those quantitative comparisons yet, so I don't have that data for you yet. Hopefully we can do that. Part of it was we had to come up with these new electrodes that were the right geometry to even get those measurements. The microdisc electrodes weren't really going to work for that, so I'm sorry I don't have an answer yet. Yeah. Okay.
Another area of importance for diamond that you did mention for neuro application is encapsulation. And I've heard from several companies, and they can't tell me anything about it, that they are using diamond as a coating for feed-through and encapsulations. I wonder if you know anything about this, and if you do, can you tell us about it?
Well, I don't have any inside tracks at companies to help you with what they're doing or not doing. In general, there has been some work in the diamond area, in the diamond field, of looking at cell adhesion and just cell viability at diamond. They do seem to show less protein absorption than other materials from what's been reported. But they also show that, that there's not necessarily, there's good cell viability. There's work involving mostly nanoparticles. They're considering diamond for a drug delivery vehicle, and so they've looked at cell viability with injecting nanoparticles, and also as a surface for growing neurons. And the cells seem to be happy there if they promote, they're still using growth factors to grow the neurons in that case, but they did see the cells were happy on the diamond. And actually, I have a little bit of preliminary evidence of growing cells on diamond, and they seem to be happy. But I think what's going on with the encapsulation is that they're promoting the idea, the diamond, the hydrogen terminated surface that you get from the diamond growth environment is more hydrophobic and is also maybe less attractive for the proteins. That may be what they're going after. I haven't heard anything else magic yet that they're talking about.
So I have one. Do you envision down the road that there may be an electrode technology that can, one material, do stimulation, recording, and chemical detection?
Yes.
What material might that be?
My material. I think diamond, I think it really, I'm not going to claim that it's going to be superior in electrical recording. It might be comparable, although the baseline current is lower, so I'm hopeful it'll be superior in terms of being more quiet in terms of noise levels. But I do think diamond could be comparable or superior to other electrodes for all three. So, and I have had interest already in trying to integrate 2 or 3 of those together.
So
properties.
From a materials synthesis perspective, what's the single biggest challenge in getting a diamond electrode to be a good material for stimulation and recording?
So I'm supposed to repeat the question, aren't I?
Did I not say that loud enough? I did. I violated my rule. So from a material synthesis perspective, what do you think is the single biggest challenge to get a diamond electrode to be a really good, comparable or really good material for stimulation and recording? Like doping or something like that?
I think that, well, one of those issues is, which is something we originally hadn't thought about, was the integration of with other materials. That is a big, big challenge in any application of diamond, especially in this one. I think the for neural stimulation, some of the advantages of diamond for sensing are maybe hurt us a little bit on the charge injection side for stimulation. So what we're wanting to do is try to preserve the advantages of it in terms of stability and versatility while enhancing the charge injection. So I think trying to combine the advantages of both. And it's, I mean, we may have an array and you have some electrodes that stimulate and some that are recording, and you've done something to specialize one or the other, but it should work being integrated.
Other questions?
One question.
Wait, Bill.
It's very interesting work. So my question is, for recording, what is the contact resistance typically required for interface with the neural cell, which is firing? And what's the required doping level to do that?
The doping level, what we grow, I hadn't mentioned the levels, it's about 1,000 parts per million boron, which is in a concentration in terms of atoms per cubic centimeter, that's about 10 to the 21st, which is semi-metallic in conductivity. It's not metallic in term, it has to be another order of magnitude to be truly metallic by definition, but it is semi-metallic, meaning the resistances are low enough to be used, and you can see that by the fact that there's no rectification. You can get, you can polarize it in either direction and get significant amount of current. There are people, of course, trying to use it as a semiconductor, so you have that. So that's what the conductivity is. I don't actually know what the contact resistance would be yet with the cell. Yes.
Heidi, that's really nice. I actually didn't realize how far you know this had come. It's really exciting, and I have to now go and read your paper. So, but I was trying to put this into perspective and think about where you know this technology might be most advantageous. So I was trying to think. It appears in terms of stimulating electrodes. Okay, it appears that, so I'm looking for sort of to try to get a broader perspective of where this might be advantageous to me. So it appears to me, as I'm thinking about this, that maybe some of the most impactful areas might be where you really have small surface area, high current density, kind of needs. Is that right, as opposed to sort of the macro electrode technology, perhaps? Would that be a sort of a first cut in? Is my assumption correct?
I think because you can take diamond to higher currents before you, say, break down water, before you maybe get to irreversible reactions, or higher potentials, I should say, that you could potentially go toward higher current applications and remain reversible, which I think is the key. But I think another aspect there is, from the integration standpoint, I think is trying to apply, say, chemical recording for feedback with stimulation. That's definitely something that I'm trying to aim for early on, is that.
Yeah, I was trying to, sort of the thing that was sort of sticking out in my mind is, wow, some of the things that you're, thinking about could really impact some of the visual prostheses, maybe as a, where I don't know what really what their problem is with current density and charge density and all. I'm wondering if
and there are two other groups in the world that are looking at diamond in some way for visual prosthesis. And one of them is based out of Argonne. They're using an ultra nanocrystalline diamond material. And then there's a group based in France that they've actually got an electrode array and polymer looking very similar to ours. They've come out pretty much just about right after we started with ours. So, and they're their entire goal of their project is a retinal prosthetic.
Okay.
So, and they do have, speaking of the compatibility, they have data showing their array, at least, they weren't stimulating, but in terms of in the tissue, it survived in the retinal. They implanted it in an eye. So.
Thank you. Other questions? We haven't received any through our online link, so if you're out there in the world, in the Ethernet somewhere, the internet, you want to fire off a question, you need to do it pretty soon, because otherwise we're going to adjourn if I can't ferret out any more from the audience. So, going once, twice, 3 times. I'm not going to go a 4th. So let's thank Heidi one more time.